Hi, so today we will be discussing one numerical from the microbial growth kinetics. This will be coming under the broad module of microbiology, as well as this will be covering your some portion of the bioprocess engineering as well. So before going into the details, let me read out the question. Two bacterial strains X and Y have doubling times of 1 hour and 2 hour respectively. If they are mixed in equal proportion, then the doubling time of the mixed culture will be 1.5 hour, 2 hour, increasing with time exponentially and decreasing with time exponentially. So before going to the details, let me quickly brush two of the initial formula that you might have learned in your microbial kinetics. If you remember, number one was ln x is equal to ln x0 plus mu t, where this was the final cell population, this was the initial cell population, specific growth rate and time. And the second formula of doubling time, that was Td doubling time is equal to ln 2 by mu, that is mu is the specific growth rate. So now let's go within the problem. So here generally when we come across different problems, we talk about a single bacterial population. Probably I think most of the numerical problems that you have done from this segment that involve a single microbial population. But here, this is something interesting, that instead of dealing with a single bacterial population, we are mixing two different bacteria and developing a mixed culture. So what to do? So let's first discuss the independent cases. Then we will be clubbing the both. So if we consider, only you have to remember that it has been mentioned that both the bacterial population have been mixed in equal proportion. This is the catch line. So let's see. So I am talking about the X bacteria at first. So the doubling time is 1 hour. So let us take few cycles of these doubling time. So this is your T is equal to 0. Then T is equal to 1, T is equal to 2, T is equal to 3 and T is equal to 4. For the Y bacteria, just to have a parity, this is T is equal to 2 and this is T is equal to 4. So now they have mentioned that both the bacteria are mixed in equal proportion. So let's consider the initial cell population of the bacteria was A. Okay, so definitely since they are in equal proportion, Y also must be A. So this is the count of, or cell count, let's say, or population, whatever you feel like. So after one cycle of doubling time, this becomes 2A, then this becomes 4A, this becomes 8A, and finally 16A. Right? For Y population, its doubling time is 2 hours. So after 2 hours, this will be 2A and this will be 4A. Right? So now what happens about the mixed consortium? So in the third case, we have mixed both the bacteria in equal proportion. That means this will be A plus A. Right? So here the initial cell population is 2A. But we do not know the doubling time of this consortia where we have X plus Y, which is a question that we need to find out about the doubling time. So let's just consider I do not know the doubling time, so what I am doing is I am just comparing with these individual cycles, right? So after 2 hours, we will be having, because here both the bacteria will be growing independently. Yes, here you have one assumption that you have to think that for the entire time span of the experiment, the culture or the media is not going out of the essential nutrients. That means any of the factor or any of the nutrients present in the culture will not be having any impact on the growth. So they are free to grow completely. So after 2 hours, we will be having actually 6A. This plus this, because this will be growing, this will be growing. So after 4 hours, after 2 hours, we will be having cumulative population of these two bacteria, which is 6A, right? Then again, after 4 hours, the cumulative population becomes 16 plus 4, that is 28. So this is what we can have. Now let's see. So ideally, for this culture, we can say the doubling time should be a time point where this population actually reaches 4A somewhere here, right? Now, again, that means it will be definitely moving towards the time of inoculation. Again, for this particular point, once we are done with our 4 hours experiment, we have 28. Now you see, this is just, if you compare these 2 hours thing, instead of this doubling thing, it has already tripled, because 2A into 3 is 6A. Right? For the first 2 hours. Now moving for the second 2 hours, what you see, if this is 6A, that means doubling time will be that time when it reaches 12A, maybe somewhere here. But after this 4 hours, it has reached 20A, means it is more than 3 times again. That means it is very clear, the more the doubling time cycles you are considering, we are having a reduction in the doubling time. So as it stands, the final answer should be, if you are having a mixed consortia, a mixed population of bacteria, the doubling time will be decreasing with time exponentially. So what is the very practical example if I talk about? Perhaps you are aware, or perhaps you have done some microbial culture where we often find contamination, right? So what happens? Suppose you are doing an E. coli culture. So if we somehow get that culture contaminated with some other XYZ strains, you will find that in a particular time, after a particular time span, the viscosity of the medium is drastically increasing. That means what? The contaminating culture is there along with the E. coli culture. If it would have been a normal pure culture, then the cell population that you were supposed to get after one doubling time cycle, definitely that will be increasing with time because there is a contaminated culture. So this is a practical example. So the take home message is, whenever you are going from a pure culture to a mixed culture, all the time the doubling time will be decreasing exponentially with respect to time. So when we discuss this sum, probably you have noticed that initially I wrote two formula, and whenever you are encountering such type of problem with doubling time, population, etc., naturally your brain will be probably moving along a direction where you might find that I have to use this formula. But this is a very interesting example that you see I have not used any of this formula, but still we can reach the answer. So these are the things that you need to take care of in while you are preparing for your gate. the stereotype thing or the normal numerical formula which are taught in the class may not be always useful while tackling some typical problems that come in GATE. I hope I am clear at my message.